Okay, now. Exactly. Let's spend a minute about like thinking about why do we need to care about what I just mentioned? Why do we need chemicals and not all? And I just want to highlight how important and how relevant these chemicals are, synthetic chemicals are in our daily life. Just think of you being sick or taking a shower. So for our health and personal well-being, we need pharmaceutics and personal care products that, of course, depend on synthetic chemicals. And then in some functions, it's maybe some of the functions of synthetic chemicals is maybe more hidden. So when you think about food production, being it pesticides or plastics that are important in agriculture, and we will come to this more specifically in a second, these are more hidden, but still nonetheless very important functions of synthetic chemicals. And then think about bottled waters and also textiles, where chemicals just played an important role. So I just wanted to start with this to show you, yes, we care about the environment, but we also need to think about other aspects of chemicals and starting from a point where we cannot simply ban just all chemicals or get rid of them, but they really play an important role in our everyday lives. However, and that's the big but here, that's why we are all here. Of course, there's problems with this, right? So chemicals in many functions, many applications can get released into environmental system. And we mainly care about natural systems such as river waters and the ocean or soils. And it has been shown that many chemicals have harmful effects on life in there, so on these environmental systems, and in the end, also on us humans who depend on these functioning ecosystems. So this poses an important and very difficult balancing act on us. On the one hand side, we need chemicals, and on the other hand side, we know that synthetic chemicals can cause harm to environment, in the end also to humans. And this is nicely illustrated also by the United Nations Sustainable Development Goals shown here. For example, we need chemicals to zero hunger and to provide good health for everyone. And at the same time, we want to do everything we can to protect clean waters and protect life in water and on land, so in aquatic and in terrestrial environments. A group of researchers then gathered together, and this was discussed in this series already, so I'm not going to go into too much details here, but this group of researchers gathered together with the goal of guiding us as humanity in this process of doing well in this balancing act. So what is the safe operational space for humans on Earth? And I want to highlight this wedge here of the circle, novel entities, where of course synthetic chemicals play an important role. And for many years, it was not quantified or not assessed whether we have already passed this boundary, crossed this boundary, or whether we consider ourselves being on a safe space. And very recently, early this year, there was this important paper which already from reading the title, you can just read the first word, outside the safe operation space. So what we need is solutions in this balancing act. We need chemicals that don't make us cross this boundary even more, or maybe, let's get back into this safe operational space in terms of how we use synthetic chemicals in our lives. And one approach towards such a solution is called environmental benign-by-design. So let's create chemicals that still serve the purpose. They need to fulfill a function. They need to be stable during use. But the idea here is that the chemical, when it gets into a system where it does not belong, for example, into a river, that there it is not stable, but rather degrades into components that are not harmful then to this environment any longer. So benign by design, and this again has many aspects, but where we focus on tonight is really chemicals that are designed to be biodegradable in the environmental system that they reach and therefore cannot cause an effect any longer, while at the same time being stable and functional during their use. And so basically what we want is shown on this picture here. We want kind of everything, right? We want to be safe and healthy and everything, but we also don't want to harm an environment. And sorry for the non-German speakers in the audience. I know it's a poor translation here. I just went word by word, but the eierlegende Wollmilchsau is kind of what I thought of when I prepared this talk. But I guess also for non-German speakers, the image is pretty clear. And so let's discuss tonight a bit, how close are we there? and what do we still need to be done so that we can go closer to this funny looking animal here. The first and very important take-home message that I already want to highlight here is that such an approach should and cannot be taken for all the chemicals that we have. You know, it's simply too many functions, and for many chemicals, it's also not the goal to have them biodegradable. So the point I guess I want to make, it's really for very selected applications where this approach makes sense. As Thilo mentioned in the introduction, biodegradable just by itself means is positively connotated. So it's kind of, yeah, we want this, but be careful. It's also being used in greenwashing thing a lot. So we really carefully think, is it really needed for this certain application to have a chemical biodegradable? And then if so, what does this really mean? And we'll come to this in a second. But for now, just remember, it's really only for selected applications where biodegradability in the environment makes sense. And for what examples and what applications are examples to these? So kind of one criteria is that it's very likely or given that a chemical during its use gets into an environment. Think about the composting bag that you need or that you use to collect your garden and kitchen waste. So basically, by the definition of this function, this plastic bag will end up first in compost, and then if the compost is applied to land, it will reach the soil. So by the very definition of its function, this plastic, this chemical gets into an environment. And there I would argue, there it makes sense to design the chemical for biodegradability. Another example, and we're going to talk about this quite a bit, are plastics used in agriculture to increase growth yields and therefore increase the food efficiency of agricultural lands. Also there, chemicals, these plastics get into the environment just by its use. They're hard to collect, and therefore it makes sense. Another example are chemicals, a large group of chemicals that, for example, pharmaceuticals and personal care products that get into the environment. Also, just by its use, we take a pill, it's not completely metabolized in our body. We secrete the chemical, it gets into the wastewater, and thereby, if not completely removed during the wastewater treatment process, into natural environments. And you can imagine how difficult it is to get out every chemical during a treatment process, even if it's a very advanced one. So also some functions of chemicals that get into wastewater are considered beneficial when we can implement the biodegradability aspect. And in the remainder of the talk, I would like to go a bit deeper into two examples that are considered meaningful, so where biodegradability can play a role. And the one on the left-hand side where I would like to start with is plastic biodegradation in soil. And then in a second, a little bit of a shorter part, I would like to touch on antibiotics that get into the wastewater and how biodegradability can play a role there. So for the next few minutes, let's talk about the process that is shown here on this picture. What you look at is a plastic surface after this plastic has been buried in agricultural soil for some weeks. So we took this plastic out of the soil and took an image on its surface. And what you can see, that is some organisms from the soil started to colonize the plastic surface. And at some spots, you can see that the organisms started already to eat and chew on this plastic. And we are going to talk about why this is good or why this is potentially good, and what kind of parameters control this process, and also what do we still need to learn about this. But let's first zoom out a bit and talk about why does so much plastic actually end up in agricultural soils every year. And so this is one function, one application. It's called plastic mulching. And I'm sure many of you have, for example, when you're on the train ride and look out of the window, you see a lot of agricultural lands are actually now covered with plastic. So the image on the left-hand side. Why is this done? I'm not a farmer, but the main goal of these plastic applications are basically to, per area, produce more vegetables, etc. And at the same time, you want to save the amount of water and pest and herbicides that you use on these lands to achieve those crop yields. So in general, in principle, this is a very sustainable technique because, you know, you need less land to provide enough food for the growing human population. Then when you look at the right-hand side of this slide, you can see that this is not necessarily a truly sustainable technology because these films, to be economically feasible, are very thin. And so after a harvesting season, it's very difficult to entirely remove all of this plastic from the film. So a lot of plastic gets into agricultural soils with every application, as you can see an example on the right-hand side. And the main reason for this is that this plastic called polyethylene, and you see the chemical structure here for those of you that are interested on the bottom right, is mainly consists of carbon-carbon bonds at the backbone. And this makes this material very stable. And so you can see, obviously, if something is stable, it accumulates over the years in a system. Just to add a scale dimension here, this is a satellite image of a southern area, of an area in southern Spain. It's roughly 200 square kilometers covered pretty much in plastic. And this is just one example. So this is really a widely used technology with a good purpose, but then with this potential problem to the environment. So what's the idea here? Can we create chemicals, plastics, polymers in this case, that serve the same function? So they're stable during their use, but then once they are plowed in, for example, when they cannot be completely removed after the harvesting season, can we design them in a way that they're completely biodegraded by the organisms in the soil so they do not have such an accumulation? And the idea is, instead of those really stable chains, to include these predetermined breaking spots every now and then into the chemical formula, so that, you know, organisms that grow on these plastics, as I showed you in the image before, that they can then use their enzymes, their metabolic machinery to break down the plastic and fully convert the plastic into its building blocks. For example, under aerobic systems where there's oxygen available, the main products would be CO2, and there would also be some biomass formed during the biodegradation process. With this, I would like to come to a second take-home message. Again, here you will read a lot in scientific articles, maybe also in textbooks, but also in the news. It has been shown that chemical X is biodegradable. This is per se good news, but only if it's really true. And I want to highlight here that really something is only biodegradable if you can show what I just explained, that really everything, all the building blocks of a material are fully converted into something that does not harm a certain environment. And only then you can talk about biodegradation. If you only monitor something and realize that the concentration of a chemical decreases over time, or that, you know, a plastic particle that you add to a soil is no longer visible after a year, that does not necessarily mean that it's biodegradable, but it's just a dissipation or a disintegration. That's really not the same thing. So be careful there. If you read biodegradable on something, really make sure and be critical if it's actually has been shown. And in similar terms, there's often a confusion in the terminology. So I would like to spend a minute here and discuss another term that is related, but means something completely different. So we talked about biodegradability means, and then there's the term bio-based. And bio-based means where do the elements in a material come from? So is it something that is renewable, for example, a plant-based material, or is it, for example, fossil fuel and petroleum-based material? And while there is plastics that fulfill both of those, so they are biodegradable and bio-based, there is a group of chemicals that do not fulfill any of both of those criteria. So they're not biodegradable and they're based on fossil fuels. And while these two groups are the most prominent ones, all combinations of those two things appear. So there's also plastics that are fossil fuel and biodegradable, and then there is plant-based materials that are very persistent and so not biodegradable. Just an important concept to really separate those things. And while both of them are important for certain applications, that do not necessarily link to each other, because the biodegradability, as mentioned before, is a factor is a function really on the structure, the chemical structure of a material, and not really depends on where do the elements in this chemical derive from. One way, now we are back, this was just a short excursion into terminology and bio-based, but now we are back at biodegradation. And I want to show you one way how this can be demonstrated in a complex system. And this is an example from my own work. It was a research collaboration between ETH in Zurich and the University of Vienna, where we wanted to show the biodegradation of a plastic, the one that I showed you the structure of before, in a soil system. So what we first wanted to see is there CO2 production? Remember, this is one of those endpoints, these measuring endpoints. Is there CO2 production if we incubate a polymer in a soil? And we used labeled variants of this soil. So certain positions in the chemical structure were labeled with an isotope so that we can more easily track them in this complex environment. And we could show for different variants, because it's, as I mentioned, important to show biodegradability of all building blocks. With different variants, we could show that, yes, actually all building blocks of this material are converted into CO2. So one endpoint we had covered. And so we took those films out after about 6 weeks and looked at their surface with an instrument called NanoSIMS, nanoscale secondary ion mass spectrometry. Not important what this is. In principle, it's a combination, in very simple terms, of a microscope and a mass spectrometer. So remember those labels that we put into our polymers? We can create an image and see where this label go during the biodegradation process. And so again, here you can see at the top, we did have some film surfaces that were colonized by soil microorganisms. But what is really important here is this bottom row of images here, where we see where our labels go. And if those organisms that colonized the films were purely grown on soil carbon, so had nothing to do with the biodegradation of the plastic, they would appear blue here. And you can see that there's a few organisms, this bacterial cell here, or this fungal hyphae here, that are indeed blue, so they probably didn't contribute to biodegradation, or at least not take up material from the plastic. But the majority of those organisms are more towards the yellow and red scale, meaning that from all the building blocks of this plastic, then there was some taken up by the organisms that colonized the film. Important here, together with the CO2 measurements that I showed you before, we really demonstrated that this plastic is in principle biodegradable in soil. And this was good news. So we were really happy that it was not only shared within the scientific community and in our research paper, but it was also picked up by news outlets around the world. So I think it's good that good news is shared, but very important in research is always to really, as soon as this good news is shared, to also put out the limitations there. And one thing that we did not show, under which time does this happen, right? We showed in principle, yes, it's biodegradable, but of course you can imagine if we care about whether a chemical accumulates in the environment or not, the time is a really important aspect. And now one could go back and do these experiments for all different combinations of soils and different combinations of chemicals, but this would take probably a really long time. So what is typically done then is, can we, study a simplified process and have methods to get to an answer much quicker. And what we did here is to focus on what is assumed to be the rate limiting step of the biodegradation. So how these enzymes that are secreted by organisms, how do they chop up the polymer into smaller pieces? And we use the method where we have a very thin layer of the plastic and incubate them with an extracted enzyme, and then through mass decrease can discover very quickly whether a polymer is degraded or not and how quickly this occurs. Of course, this is a limited system. It is by far not as complex as the soil experiments I showed you before, but still you can really quickly get answers to what factors affect the biodegradation of a polymer. We tested a bunch of factors, for example, environmental conditions such as the pH or the temperature. How would this affect the rate and extent of polymer biodegradation? But I want to show you some highlights on how the composition of a material and how then the enzyme that plays a role governs this process. And so we stick with the same polymer for simplicity here. And we, again, without going into too much detail, what we had is 6 different variants of this polymer where we changed the composition of 2 different building blocks. So the red ones, the red curves that you can see here, contain more of this half of the molecule, and in simple terms, and when we go towards blue, we add more of this component. So it's the same chemical structure, but the composition is slightly better. And you can see that it has a strong effect on how quickly this plastic is degraded by the enzyme. So we use a cutinase from a soil fungus. So yes, that's no surprise, but the chemical composition of the material plays an important role in governing how quickly does this process happen. What was maybe a bit surprising is kind of how different that was for another enzyme. So here we did it then again for a lipase, another group of enzyme from, again, a soil fungus. And you can see that not only does it get slower, but it really does, there's no biodegradation at all any longer if you go towards more of this compound. So what this means is not only does the material determine how quickly and whether something is biodegraded, but also the microbiology of a system has a great effect. So it really matters which organisms are there and which enzymes are in an environment when a chemical is released. And so one big open question in this first example that I showed you is which organisms and enzymes do really do this job under environmentally relevant conditions? For example, in the soil in this example. Yes. And to highlight this point even more, I want to mention a study that was done a few years back where they took plastic bags that were designed to be biodegradable in industrial compost, so kind of compostable bags, and they incubated them either in seawater, in all different kinds of combinations of environments actually. But here, the images I showed you were incubated either in seawater or in soil. And what they found is that these compostable bags were pretty much intact still, so not really biodegradable during 3 years in these environments. Based on what I told you before, you should not be surprised right now, right? So the finding was basically if you take something that was designed for biodegradation in one system, if you take it into a different system, you don't know what the outcome is. So I think that was an important study, but unfortunately it was misinterpreted by quite so many people, which then concluded, okay, biodegradable plastics are actually not even biodegradable. So why should we even care about this and just move back to conventional products? And I think this is dangerous, right? We need to really be clear in communication what these things can be good for and under what conditions they are actually biodegradable. So there is probably no such thing as a biodegradable plastic for the environment because it really is environment specific. With this, I would like to shift gears and come to the second example where I think biodegradability of a chemical compound is desirable or at least worth checking for. And this is antibiotics that are released into wastewater systems. I don't think we need to spend a lot of time discussing why antibiotics are important, of course, and hopefully not too many of you, but probably a few already depended on antibiotics when they had a bacterial infection. And so there's currently a great interest in novel antibiotics, also driven by this resistance formation that is going on. So it's expected that these pathogenic infections will be a major cause of death, the bacterial infections, also because there's just so many bacteria that are resistant to many of the antibiotics that we currently use. So this is a very dynamic research area, the development of new antibiotics per se. And I think a small perspective there from the environmental side could also help in the process of developing new chemicals for this purpose. I mentioned it already in the beginning, but here maybe again, why do antibiotics get into wastewater in the environments at all? Well, if we take them, being it orally or maybe intravenously into our blood, our body does not really metabolize all of it. So a lot of the chemicals that we take are secreted, for example, through urine, and then get into wastewater. And this has been investigated in a study that was published early this year. Also, researchers from our department, Thilo Hoffmann, was involved in this study. And what the researchers here did is they collected samples from more than 130 water samples all across the world. And then they did targeted analysis. So they went and for a set of compounds, for a set of chemicals, they wanted to determine their concentrations in all of those water bodies. And I highlighted here with some red arrows the antibiotics that were included in this study. And what you can see here for these chemicals, as for the others, of course, the concentrations determined in these different water bodies differed from each other, but you can see that in most of those analyzed samples, there was quite high concentrations of all of the compounds and also of these antibiotics. So why do we worry about this? Of course, antibiotics, you know what they do. They can kill bacteria, and many of the ecosystem functions that we depend on, of course, depend on microorganisms. And also low concentrations of antibiotics can change the community composition of these microbial ecosystems, and therefore can have really bad effects on the functioning of the environment. And of course, another thing that can happen is that also these environmental microbes, they form resistance. So resistance genes are spread within those systems. And this does not only happen at clinical concentrations of antibiotics in our body, but it can also happen, for example, in systems like wastewater or even downstream environments that are exposed constantly to antibiotic concentrations. So what we definitely need, in my eyes, is a better understanding of what happens to antibiotics on their way into the environment, in wastewater, during the wastewater treatment process, and if needed, also in river systems. So what we did here is take a small set of antibiotics. We went to a couple different wastewater treatment plants. This was work that I did back in the US. And then we incubated those together. So we added some antibiotics to the samples we took from the wastewater treatment plant and wanted just to know how quickly do they degrade and what are products of the degradation. So on this slide, I show you the kinetics part. So how quickly did those chemicals that we studied disappear during those incubation experiments with wastewater microorganisms. So darker colors here are a fast removal, whereas lighter colors, white, mean no removal at all. And you can see by just focusing on the left-hand column for the three wastewater treatment plants, you can see that there's a very big difference across the different antibiotics in terms of bio removal. So some are removed very quickly and some others are pretty much stable. And interestingly, this was the same for these 3 treatment plants. The rank order was exactly the same. Of course, this study would need to be put on a much larger scale to really get to big conclusions, but just from those that we studied, it seems to be relatively conserved which antibiotics are removed quickly and which ones are more persistent. But what really matters here is what is the reaction product, right? It can be that a compound is transformed into something that still has the antibiotic function. And so in terms of resistance formation, etc., this would not solve the problem. So what we did, particularly for the ones where we had rapid removal, we wanted to know what products are formed. And without going into detail and sparing you all these chemical structures, I just want to point out that we found one chemical bond that is very often cleaved, and that's this amide bond here that is highlighted by the red circles. And so this is maybe for some of you with more expertise not very surprising, because of course in wastewater there's a lot of enzymes that can catalyze this bond. For example, proteases that play an important role in the protein metabolism. So there's a lot of protein in the wastewater that gets in there. And so organisms that want to degrade this protein, they would secrete those enzymes. And then maybe in a process called co-metabolism, those enzymes can also transform our chemicals of interest, in this case, the antibiotics. And so another thing that I wanted to share with you is what we, a very recent finding in my group from Natalie Wichmann, and what we found is that these peptidases are not necessarily only in these bioreactors at the wastewater treatment plants. So typically biotransformation studies, such as the one that I showed you just before, are typically done in these tanks where in these biological treatment steps. However, what we now did is for different wastewater treatment plants around Vienna, we went at different treatment steps. So also, you know, just after the big chunk is removed after this initial screen, kind of the influent concentration. And we also looked whether there is enzymatic activity there, the one that we're looking for. And so throughout the treatment process steps. And what is interesting for these peptidases that I mentioned to you before, and where we think that they have quite some potential in transforming synthetic chemicals, the actually highest activity was found to be in the influent. So these are probably not enzymes that are produced by organisms that live in these bioreactors in wastewater treatment plants, but rather enzymes that get into the wastewater treatment plant together with the chemicals in the wastewater. And so I think taken together, this susceptibility of these amide bonds, this chemical bond that I highlighted before, and the high activity of peptidases in influence, we think there's a lot of promise also from an environmental perspective for a group of antibiotics called peptide-based antibiotics. Of course, environment is only a small aspect when we think about antibiotic development and importance in general. So it's kind of great that this is a dynamic field of research anyway. Every year you have a couple of really high impact publications in great journals that show a new chemical from this class, a peptide-based antibiotic with a lot of promise. And we think that also in terms of environmental chemistry, these compounds might be very interesting to follow on. However, again here, we are just at the beginning. So what we of course then need for each compound is really to understand what happens. Is it true that also in this new compound, this bond is cleaved and leads to an inactivation of the antibiotic? Another big question is then, what does this actually mean for resistance formation? You know, it's not necessarily given that a biotransformation, which leads to the inactivation, for sure reduces the resistance formation. For downstream environments like the river system, that's true, because if there's no antibiotic that gets in there, there will be less resistance formation. But for the wastewater treatment plant itself, that's to be discussed. And I'm very happy to discuss this aspect later in the Q&A if someone is interested. I just didn't want to go too deep into this resistance evolution in the general part of the talk. And another big question, again, as for the plastics that I mentioned before, it will be important then if we find some biodegradation reactions to know which organisms and which enzymes are relevant for this process. Also then to think about, is this a process that happens only in the very local communal Viennese wastewater treatment plants, or is this maybe something more global? So is it distributed all over the place, all over the world? And can we therefore make use of this function in the design of sustainable chemicals? With this, I would like to slowly come to an end and wrap up, and wanted to highlight this. These were two examples where I showed you, on the one hand side, the biodegradable plastics in soil, and on the right hand side, the antibiotics that might be biodegradable then in the wastewater and wastewater treatment plant. And here I want to highlight, of course, there's more examples, and we can maybe also discuss later what other examples are there. But again, reminding you that, you know, it needs to be only for very selected applications that biodegradability is really an asset for a chemical. So I would like to leave you here with these take-home messages that we mentioned during the talk. I would just like to reiterate them, and so that you can really, that hopefully everyone can take something away from this talk and the discussion. So I mentioned to you, and maybe you agree or not, and if you disagree, please raise this point in the discussion afterwards, that biodegradables, yes, they are beneficial, but only for selected applications. You know, there's other applications also in the plastic sphere where recycling, for example, is a much better option than designing for biodegradability. And this is important to consider, that yes, biodegradability sounds and is beneficial, but it's not the solution to all the problems that we have with chemicals in the environment. Then I try to define, and hopefully you have now a better understanding of what biodegradability actually means. So it means that really all building blocks of a compound are transformed into something that does no longer harm the environmental system, and that this also needs to be demonstrated. And I showed you an example of how this can be demonstrated. And then maybe the most important one, just please remember that something like biodegradability in the environment does not even exist, because you know the environmental systems are so diverse that really this is not possible. So a biodegradability claim is only meaningful if also the environment, the environmental conditions, and the time span needed to be biodegraded are also defined. With this, I would like to thank you very, very much for your attention. I'm very much looking forward to this discussion. I would like to thank the Environmental Research Network once more for organizing this great series, bringing together people with different backgrounds, different expertise, but all with the goal of learning more about environmental systems. And I would like to mainly also thank the people I work with here at the University of Vienna at EDGE, and particularly my research group here, the PhD students, Natalie, Aaron, and Richard Schoen, with whom it's a lot of fun. If you have questions, I'm very happy to take them now. If you have them later, you're also happy to send me an email or contact me through other means. Thank you very much.